Hello, how are you all doing? I'm doing fine, thanks for asking. My name is Mr. Leary, and I'm a biology teacher at Beach High School. Today, we're going to start on a topic called genetics. But before we even start on that topic, I want to say thank you for joining me. All right, so before we even dive deep into the topic and I give you any information, let's just say this. Let's say you have two couples, real life example now. Let's say you have two couples, right? One couple, I'm sorry. Let's say you have one couple. That couple consists of an African American male and a Caucasian female. That couple get together and they want to create an offspring. Offspring is a baby for anybody that had trouble thinking of that, okay? All right, so let's just say these two decided to get together and they wanted to make a baby. What color do you think the baby would possibly be? All right, so in science, we always use keys, and we use keys to let everybody know exactly what we're going to be doing and to identify whatever it is. So let's just say the African American male is represented as two capital B's, right? This is for that male. And the Caucasian female is represented as two lowercase b's, right? So these two wanted to come together and make a baby. So in genetics, what we tend to do is multiply these two together. All right, so they're going to cross over. So let's just say we end up drawing a box right here. I know that some of y'all right now, you're thinking, why does it look like we're doing math? Math and science ties in a whole lot. All right, but now, so what color do you think the baby would possibly be? I know a lot of y'all, the first answer that you're thinking is mixed, right? But is mixed really a color? I'm not saying you're wrong, but is mixed really a color? So you got a chance that the kid would be black. I'm going to put a space right here. You got a chance that the baby would possibly come out looking white. And then you got, for those that said possibly mixed, right? All right, so we got three different possible colors that kid could possibly be, right? All right, so now let me go ahead and veer back and show you all something. We're going to go over a few terms. So this is genetics, and on the front portion of it, this is a guy named Gregor Mendel. Gregor Mendel was the person that introduced genetics. So genetics is the scientific study of heredity. Basically, it's nothing but the genes and traits that comes within you that is passed on from parent to offspring, all right? Then you have something called alleles. Alleles are the forms of a gene or factor. Genotype, phenotype. Those are two different things that you can easily get mixed up. But I'm gonna teach you an easy way where you can remember it. You have the genotype, is a combination of alleles an organism has. But just think, gene is inside of you. You can't see genes with the bare eye. You can't see genes physically. But a phenotype, PH, physical. Phenotype and physical, that's the physical appearance of the organism. You can always see the physical appearance, okay? Then you have something called dominant. Dominant is, let's take, for example, a relationship. In that same couple, you have a male and a female, right? One of them is going to be more dominant, more controlling than the other person, right? So that's what dominant is when it comes to genetics. Dominant is the one that's more controlling. It's the one that's more expressed. And also in that relationship, you have something called recessive. Recessive is passive. Anybody that's a passive person, a lot of people see them as pushovers or somebody that really won't speak out and voice their opinion and tell how they feel. So that's what that recessive allele is. The recessive allele won't be expressed physically. It'll still be inside the body as a gene or a trait, but it won't be expressed. It'll be masked by that dominant trait. Then you have something called homozygous, right? Homozygous and heterozygous. Easy way to remember that. 1 second. So we're going to move off to the side. You have homozygous. You have heterozygous. Now, tell me two things that these two words have in common. You're absolutely right. Zygous, okay? Those are the two things that they both have the same. So, in this instant, we're going to say zygous means traits, all right? Zygous means traits. Now that we have these two right here, meaning the same, we're going to give them the same shape, same thing, same shape. Now you have something called homo. First time you hear homo, what do you think? Exactly. I already knew you all knew this. You're very smart. Same. Homo means same. All right? So, for homozygous, whenever you hear that, first thing you're going to think is same traits. Okay? Make that bigger so you all can see it. Homozygous is same traits. So, if homo means same, it's only fair that hetero means different. Different traits, all right? So homozygous is the same trait. Heterozygous is different traits, all right? Now that we've covered the key terms of genetics and what it means to build a Punnett square, the Punnett square, basically, the Punnett square is basically this box right here that looks like we're finna do some multiplication. That's what the Punnett square is, okay? The outside will always, we're gonna label the Punnett square with the outside as parents, and the insides will be the possibility of what that child genes and traits could come out to be, okay? All right, so, like I said, this male African-American male that's in a relationship, we're gonna put him on top. We're gonna say the man's run everything, okay? We're gonna put the females on the side, right? So, each trait, each allele will get their own box. I have a capital B, a lowercase b, and a lowercase b. The reason why I did that, now, we have two B's. Those two capital B's are the same, right? So that would be something called a homozygous trait. They both are capitals. Both of these right here are capital, and both of these right here are lowercase. So they both will be homozygous traits. But what will start to show difference is homodominant and and homo-recessive, okay? All right, so that's gonna show the difference. Dominant, like I said, dominant is something that stands out, that takes control, that masks everything. Capital letters, all right? So that capital letter will always stand out. So the capital B's will be homozygous dominant. Homozygous recessive would be the lowercase b's because they're small. This is like, let's just say a fight. A big guy getting in a fight with a small guy. Natural outcome of what everybody think would be the big guy winning, right? All right, so the big would be dominant. It will win every time over the recessive trait, all right? So, I'm gonna jump back one more time. Now that we've covered the terms, I'm gonna introduce you to Gregor Mendel. Like I said, Gregor Mendel was the founder. Well, let's say he was the father. He helped discover the reason of genetics, and he used these principles in the early 19th century without all the scientific equipment that we have today. And he did it using pea plants. He used the pea plants for crossing the color, the shape, the height, and the width of it, all right? And the color of the flower, okay? So the the pea color could either be green or yellow. The shape that he was using could be round or wrinkled, or the stem or the flower could be purple or white, and the plant could either be tall or short. He crossed those two together to see if the genetics from those parent pea plants would go to the offspring. All right. So if you look at the picture right here on the right for Mendel's experiment 1, you see that on the left is a homozygous dominant trait. A homozygous dominant pea plant, which is 2 capital S's, and on the right side you got 2 lowercase s's, which is homozygous recessive. Now, if we look at it, what's the shape of the homozygous dominant one? You're right, it's round. What about the homozygous recessive? Exactly, it's wrinkled, just like it said on the left side. Okay, so now if we move down to F generation 1, that means the pea plant times itself. That's what it came out to be, and then that's F generation 2. Basically what they did, They did the same thing that I'm finna show you right here in this Punnett square. They took, just like how math is, each column, whatever's above that column goes down on each one. And then whatever is on that adjacent row goes right beside it. So, and you will always put the dominant traits first. So right here, I'm gonna say a capital B, a capital B. Like I said, whatever's in above that column goes straight down. You're gonna use another capital B, another capital B, because there's only capital B's at the top of that square, right? Now we're gonna work on the recessive traits. Recessive are the ones that's on the side. It'll move them across, okay? You have a lowercase b in both of them. And now it's the same thing, because guess what? It's homozygous recessive trait, so it's gonna be the same thing in each square. So now, the offspring is something that we've never seen before, right? We got a capital B and a lowercase b. Now, the chance of this offspring coming out looking black, mixed, or white, if if homozygous dominant means black, and then homozygous recessive means white, we don't have a color right here, right? I'm guessing you all thinking, oh yeah, he's mixed. I guess you can say that kind of, but let me go ahead and show you something. Like I said, dominant traits will always match over recessive traits. So what color do you think will show more? The capital B or the lowercase b? Remember, the capital B represents his black. The lowercase b represents his white. Exactly, so the baby would be black. That would be the physical appearance, all right? So, like I said, now you have something called genotypes. And then you have something called phenotypes. Genotypes are the genes that's inside the child, and the phenotypes are the physical expression, like I said before. But before I show you more about this, just thought about it. We missed identifying this. Heterozygous, we think back from earlier, it's different traits. We got a capital B and a lowercase b. So this right here is a heterozygous trait, all right? All right, so that's a heterozygous trait. So now, what we're gonna do, we're gonna find out the gene ratio and the percentage that the child will possibly come out. So we're gonna take genotype, we're gonna write it right over here, right beside it. We're gonna put the ratio, small, and we're gonna put the percentage. Now, the order that we're gonna go in is homozygous dominant, homozygous recessive, move it over, and then we're gonna go heterozygous for the genotype. So, of this square, what's the chance that you'll get a homozygous dominant? Ratio is whatever it is out of 4. Exactly, 0. You see no capital B's, no homozygous dominant traits in these 4 squares. What about homozygous recessive? Right again, 0. So how many heterozygous traits do you see in there? Exactly correct, 4. So if we're doing the ratios out of 4, what's 0 out of 4 for a percentage? 0. And since we said that for the first one, it has to be that for the second one again, because that's 0. So what's 4 out of 4? Exactly 100%. So 100% of the offspring would be a heterozygous trait. That will physical appearance be black, but it will always have the gene of having the white color shown inside the genes. So now we're going to do phenotype. We're going to do the ratio again. And then we're going to do percent again, right? So we got the ratio on top, we got percentage on the bottom. This time we're going to use the colors. What's the percentage that the colors will be black? Percentage that the colors will be white. Okay? So, of that same square, remember now, Heterozygous, it's a heterozygous trait that's inside the square. Heterozygous means the dominant trait mask over the recessive trait, okay? What's the ratio that the kids will be black? Exactly, 4. What's the ratio that the kids will be white? Exactly, 0, okay? Percentage, once again, exactly, we have the same numbers from earlier, right now, I know it. But now we're gonna switch it up a little bit, all right? Let's say one of these same kids came out, right? And now they wanted to make a child of their own. So this child right here, and let's say they end up breeding with somebody that's just about like them, right? So 2 heterozygous kids end up breeding with each other. Always gonna draw a Punnett square. Got our key right here on the left. Capital B, lowercase b. Right? Now, I know in the first one, I said the capital B, we're going to put that on top, but really it doesn't matter. You can put whatever parent on whatever side. So, in this instance, you'll never know which one of them will go where, right? We're going to do it like this. One parent's up here. We're going to call him John. John's at the top. And then Mary's right here on the side. Once again, like I said, each allele will get their own box, okay? Each one will get their own box. In the first box, now what will happen? Exactly. You'll have a homozygous dominant trait. 2 capital B's will go in the first box. One go down, one come over. Let's go to the second box right here. What'll happen? Like I said, every time you put the dominant trait first. Capital B. Now we're going to bring over that recessive one, okay? Now we got one that's just like the parents. Come over here. What's going to happen? Exactly, that dominant trait is going to go first. I'm going to bring him right back down. Capital B, lowercase b again. Same way as this one. Now what's this one? Yeah, I know it don't matter. It's 2 lowercase b's, right? All right, so now we got a different combination inside of here, right? Exactly, we got a homozygous dominant, we have a homozygous recessive, and then we got 2 heterozygous traits, right? Okay, so just like always, we still got to find a genotype, then we got to do the phenotype, okay? So, genotype for this one. Homozygous dominant, homozygous recessive, and heterozygous. Okay? Now, what's the ratio of the four? How many of them is homozygous dominant? 1. How many of them is homozygous recessive? 1. Now, how many of them is heterozygous? 2, exactly. Now, anytime that you feel like you're having trouble with trying to find out which ones go where, if you end up getting to your ratio, just add it back up. It should always equal how many squares you have. 2, 3, 4. 1, 2, 3, 4. 4 boxes, okay? Percentage. Your percent will always equal up to 100%, okay? So, again, homozygous dominant. How many of those? You say it's 1. Always remember it's out of 4. So, whatever it is out of 4, just take that, whatever number from the ratio, put it on top of this right here. That'll give you your percentage. Or it'll give you your fraction, and then from your fraction, you turn it into a percent. All right? So, percentage of homozygous dominant, 25%. Exactly, so 1 again, 25% again. Now, what about for heterozygous? Exactly, 50%. Now, if you add it up, 50 plus 25 is 75. 75 plus 25 is what? 100. Exactly. So now that we have the ratios and the percentage for the genotypes, let's do the phenotypes. Okay. Now we got the phenotypes. Again, I'm going to go over. How many kids would possibly come out being black? How many will possibly come out being white? Okay? Now, got the ratio again. Right up under that, we got the percentage. Remember, percentage, in this case, is out of 4. All right? So, if we look back at the square, how many kids will come out being black? Remember now, it depends on that dominant trait. How many of them have that dominant trait? 3. And if you kind of get stuck and you kind of get a little confused, if you know this is 3, just subtract it from the amount of squares that's in the box. In this instance. 1. All right? Now, what's the percentage? 3 out of 4 is what? 75. 1 out of 4? 25. Exactly. All right? So, now, in this instance, you're like, but that don't make sense. But if you look at it, like I said, homozygous dominant, heterozygous, but you got 2 capital B's, right? So now they make 3 of them being black, and then you got homozygous recessive, like I said before, right up here. It means that the child will possibly come out Caucasian, okay? All right, so now this is what I want you to do. Now that we've done one, I want you to try to create your problem, create your scenario, and I want you to try to do it on your own, and then show me how you did it. All right, now I'm gonna come back. Now, after that, we're gonna jump back a little bit, and then I want you to tell me, now I'm gonna explain to you the laws of segregation and the law of independent assortment. So, when gametes, the sperm and the egg, sperm is the male having sperm, the egg is from the female, all right? Those are 2 different gametes. They're each formed with a recessive, to receive one allele or the other. The law of independent assortment are 2 or more alleles will separate independently of each other when the gametes are formed, all right? So basically what it's saying, males will always have a sperm. If you're going to have a girl, you're going to have a female, she'll have an egg, all right? After that, 2 alleles will separate independently. That's just to sum it up real easy. Now, like I said, the Punnett square. Hopefully your Punnett square end up drawing and looking exactly just like this, okay? And I always make that square, you'll always just draw 1 square, and then you'll divide it in half 2 times, okay? Horizontally and vertically. And if you're looking to see why they put those 2 lines outside that square, it's just to separate those 2 alleles. That's all that it is, so that you don't get confused. Now, I hope that you did your problem just like this. When you use your example, you can use any letter. That's just like for his. He used T, but he was doing height. So for tall, he used the capital T, that's a dominant trait. And then for shortness, he used small t, okay? So if he had a homozygous tall plant, like I said, the capital T is tall. That's homozygous dominant, 2 capital T's. If he crossed that with a short plant, what would that be? You're right, it's a homozygous recessive, but what's the letters? Exactly, 2 small t's. Now, what he's asking is, what's the genotypic makeup of the offspring? And what's the phenotypic makeup? So we're gonna do it with him. He said 1 plant. T equals tall, capital T, lowercase t, short. right? That's what he said. Then he said he's going to make a tall plant, a short plant. I'm going to draw the same square that he drew. Draw it outside just like this, and I'm going to show you why we drew it on the outside. Now, just like him, we're going to label it, capital T, capital T. That's why it separates it. That's why he drew that line outside, so it can separate it, so you don't get confused. On the bottom, lowercase t, okay? Let's see what he has to say next. See that? Boom. We already knew it, and we already had it before him. So he said determinate alleles of each parent. These are given as capital T's and lowercase t's respectfully. Then he said take each possible allele as the parent, separate them, and place them into the alleles. We already did that. We're already a step ahead. Now, what Gregor Mendel wanted us to do, he wanted us to see how he ended up bringing everything down, finding the possible outcome of the offsprings. So, in this same instance, like I said, you always bring that dominant trait into the square first. Right, each one will get a capital T. Then what'll happen next? Exactly, each of them will get a small t. Because we have two homozygous traits, two homozygous parents. The genotypes, phenotypes. Now, this can be done with anything, whether it's hair color, height, color, eye color, length of your fingers, anything. Even size of your foot. This could go for anything. All the genes, all the traits that you have in you, they will always come to the genetics. And it's like in small little Punnett squares, okay? So, get that ratio again. And let's get that percentage. Now, we're going to set it up the same exact way he did. Oh, okay, since he left us to do it, we're going to go ahead and do this one. All right? So, with the genotypes, exactly 100% of them is capital T, lowercase t. So, are the plants going to be tall or short? Tall. Exactly. All of them or just a few of them? Exactly, all of them. All right? All right, so now that we saw this, I did it, we did it. Now, I want you to do one on your own, and I want you to bring it back to class tomorrow. All right, thank you.